So hi, my name is David Walker. I'm a postdoctoral research associate at the University of Glasgow, and my project is investigating the short and long-term effects of novel radiotherapy and drug combinations on the normal brain using both cellular and in vivo models. I'm a neuroscientist by training. I got my PhD in neuroscience from the University of St Andrews, and the current role in Glasgow is my first postdoctoral role. And my long-term research interests are in neuroinflammation, particularly within disease phenotypes and normal tissue settings. So I am in the research group of Professor Anthony Chalmers, whose ultimate research goal is to identify novel radiotherapy and drug combinations designed to sensitize brain cells to radiation, to sensitize cancer cells to radiation while sparing normal tissue, ultimately to improve the outcomes for patients with brain tumors by overcoming the inherent resistance seen in cancer cells to radiotherapy. So the Incucyte gives us the opportunity to screen novel drugs in a high throughput manner, which allows us to investigate not only the effects that these drugs are having on our cells, but also in response to other experimental stimuli such as radiotherapy. We use the Incucyte to do high throughput analysis of cellular responses to novel radiotherapy and drug combinations in a variety of different cell types. And the high throughput nature of this, and as well as preserving the cells in their ideal environment without any disruption, is really what the Incucyte is beneficial for. I want to particularly thank my supervisor, Professor Anthony Chalmers, but also my collaborators in the lab, particularly Rodrigo Gutierrez, who's a PhD student working on the same project, and Karen Williams, who's also heavily involved in the project, David Strachan and Leo Carlin of the Beatson Institute for allowing us use of the Incucyte, and as well as a couple of people from Sartorius, particularly Matt Tomlinson, who helped me with the image processing. So just a bit of background research about the lab. So the Chalmers lab is interested in the effects of radiotherapy in combination with drugs as a way of treating glioblastoma. So glioblastoma is the most aggressive primary brain tumor and is particularly invasive, meaning that high doses of radiotherapy are often delivered over a large volume of tissue. And as a result of this causes a bleeding effect into the normal tissue, resulting in some of the irreversible long-term cognitive impairment observed in almost 50 to 90% of patients. And currently, the current modality of treatment is 6 degrees and 30 fractions with temozolomide. And however, the precise mechanisms of radiation-induced neurotoxicity are still being heavily investigated in ways to mitigate some of these devastating effects. So it's hypothesized that in the normal brain, radiation damages neural stem cells, particularly in the neurogenic niches of the brain, such as the subventricular zone and the dentate gyrus of the hippocampus. And specifically, it damages the DNA within these neural stem cells. So the research goals of the lab are to explore the cellular responses to ionizing radiation in neural stem cells and in GBM cancer cells, and then we also evaluate the safety and efficacy of drug-targeted therapies to identify novel radiotherapy and drug combinations that are designed to sensitize cancer cells to radiation while sparing normal tissue, with the ultimate aim of improving the outcomes for patients with brain tumors by overcoming the inherent resistance seen in cancer cells in response to radiation treatment. So in the lab, we've been using high-throughput live cell monitoring and imaging of these radiation drug combinations using the Incucyte. And the Incucyte is really beneficial because it allows us to screen multiple drug compounds and multiple dilutions in various cell types at any one time in a very high-throughput format. And specifically in the lab, we've been looking at how our radiation drug combinations affect cell proliferation and investigating the mode of cell death, where the cells are dying from apoptosis or cytotoxicity. I'm going to be talking about mainly the effects on apoptosis. So for the apoptosis, we've been looking at an exon 5 expression using Incucyte verified an exon 5 conjugate dye. So dying cells expressed this phospholipid residue, phosphatidylserine, on their cell surface when they're dying, and the exon 5 reagent is known to bind really strongly to this residue. And in doing so, it can light up our cells when it's tagged with a fluorescent marker. And this is a sort of typical plate map that I've been using in my experiments, which demonstrates the high throughput capacity that the Incucyte can do. So in this particular example here, we have our cells, 3 different passages of cells which are representative of individual biological replicates. We can add these to our 96 well plate format with drugs treated in duplicate, and also across multiple dilutions. So this really demonstrates nicely the high throughput format that we can do with these experiments. So with regards to the actual experimental parameters, we can plate the cells out at a relatively low density to account for the proliferation capacity. And then we can add the drugs and our annexin 5 conjugated dye 24 hours post-seeding. Then we can initially monitor the proliferation of the cells after the addition of the drug. And then after around 2 to 4 hours post-drug treatment, we can then expose the cells to either 0 or 5 gray of radiation. The cells are then returned to the Incucyte, and we can capture the proliferation and apoptosis every hour up until around about 72 hours post-treatment. So in terms of the image processing, this is essentially what the Zoom software that we use for processing the images. And using these various parameters, we can define define the annexin 5 signal based on the analyzed parameters channels here on the left-hand side, and also define the size of the cells. So this is at an early stage of the experiment here, where the cells are seeded quite lightly, but you can see there is quite a high degree of cell death in this example here. So the red dye that we're seeing here are cells dying from apoptosis, so these are expressing annexin 5. The green dye is the cytotoxicity dye that we've actually added to these cells as well. So another good thing about the Incucyte is that we can image for multiple processes at any one time using different fluorescent dyes. So this is just the image processing using the mask parameter to basically tell the software where our cells are and where our apoptotic or cytotoxic signal is here. And this is just a bit of information about some of the background correction we're using. So in this particular example, we're using the top hat background correction here, which is determined by estimating the, which is determined by estimating the radius of the most fluorescent object. So in terms of how the data, what I do with the data once I receive it all, we plug in all these values here, and then we run this analysis channel. And for a high throughput experiment such as this can take up several hours, but we tend to just leave it to analyze overnight and process all the data. The data are then exported as a TXT file. Oh, sorry about that. The data are then exported as a TXT file and then imported into Excel. And we do what's called an area correction on the data. So we do that by dividing the red and green or red signal values by the corresponding cell confidence values to basically correct for the proliferation of the cells during the experiment. And we do that for every single time point. And then we normalize the area corrected data to the first time point of data collection to observe the rate of apoptosis over time and also the rate of cell proliferation over time. So we end up with essentially raw data, which is what we get from the Incucyte, the area corrected data, and then the normalized data. And the normalized data is what we present in our presentations and in publications. So just to give you an example, this is a cell confidence data. The panel on the top, the graphs on the panel on the top there are the raw data. So these are the plots generated from the Zoom software. These have just been replotted in the R software. And then on the bottom is the normalized data. So you could see that the value for the first time point is always going to be 1. And so you can actually see the rate of proliferation over time with all these different drug doses here. The same is done for apoptosis as well. So this is done using the total integrated intensity metric from the software. So the raw data again is just displayed at the top here, and then we correct for area and then normalize. And this is the data on the bottom that which we will then use for our reports. Of course, in experiments such as these, it's always important to have nice controls. So to make sure that we're seeing true results, I've included actually 3 different controls in this experiment. So we have our dye only, where we don't add any drug to the cells. We just add the fluorescent dyes. Then we have our DMSO, which is a control for our drug. And then we have our positive control for apoptosis, where we add staurosporin at 10 nanomolar. And you can see here we're clearly inducing apoptosis at quite a high degree compared to the DMSO vehicle control and the dye only, particularly evident when you normalize the data. Okay, so this is an animation which provides an example of how the cells are growing in real time. So this has been and the corresponding graphs to go alongside it, and the graphs have been generated in R, and I have code for this if anyone's interested in it and using it. On the left-hand side, we've got the rate of apoptosis happening in real time. So the left-hand graph is the 0 gray plot. The graph on the right-hand side is the 5 gray, which is the irradiated cells. And then on the right-hand side, we have representative images, which are playing as videos, which show the cells growing in real time. And the red dye is our Annexin 5 fluorescent dye. So this is comparing our vehicle, which is DMSO, with 1 nanomolar of our drug, which is an inhibitor of the DNA, of a protein kinase involved in the DNA damage response. So if we take a look at the 0 gray plot on the left-hand side, you can see we actually get some single agent activity of our drug in protecting from apoptosis endogenously, which is nice. But more excitingly, in the presence of radiation, we actually get protection of these neural stem cells. These are not cancer cells. These are our model of the normal brain. We actually see a protection from radiation-induced apoptosis, particularly at the low doses of our drug at 1 and 3 nanomolar. So this graph here is just the same as the animation played before. This is static, just to show you give you a better snapshot of the data. And the data are analyzed using a repeated measures analysis of variance, measuring the effects of radiation and drug on our cell dying from apoptosis over time, with pairwise t-tests to compare the effects of our drug versus the vehicle. So that's comparing, in this case here on the right-hand side of the plot, it's comparing the green and yellow lines against the blue line, which is our vehicle. So you can see that at these low doses, we are protecting quite late into the experiment from radiation-induced apoptosis, but we also get this nice single-agent activity of the drug as well. This is cell proliferation data using the same drug. So it's plotted exactly the same way, but obviously we're normalized for proliferation. So at 0 gray, we actually get a little bit of enhancement at 1 nanomolar of our drug compared to DMSO. So again, the asterisks are comparing this yellow line to the blue line here. But interestingly, at 5 gray, we don't see any detrimental effects of our drug on cell proliferation. So that's good to know. We know that our drug is protecting cells from radiation-induced apoptosis, but not at the expense of the cells being able to continue to proliferate and divide. So the drug we're actually using is being used in clinical trials for GBM right now.
Okay.
And we have tested it in cancer cells. So the good thing is that it's actually sensitizing the cancer cells to the radiation as well. So we've got this nice effect of protection in the normal brain and no protection of the cancer cells against the radiotherapy when you add the drug on, which is really nice. So we're complementing this work with in vivo work as well. In animal models as well. So the end goal of this would be to identify the drugs which are designed to protect the normal brain but also sensitize cancer cells to radiotherapy. And the goal would be to continue this and to test in animal models to see if we observe the same effects, with the ultimate aim of then being able to develop these drugs for clinical use.